we have to discuss about the fluidity of cell membrane. How the cell membrane fluidity is maintained and which factors will affect the fluidity of the cell membrane. So going into the fluidity of the cell membrane, you should know that between the tails, these are the tails of cell membrane, which these are the tails of cell membrane which are made of the fatty acid, which are hydrophobic non-polar. Between these tails there will be some molecules of the lipids. Assume these are the molecules of the lipids, these which are intercalated between the tails of the phospholipid molecule. These are outside as well as inside leaflet. In both leaflet there will be some lipid molecule which will be intercalated between the phospholipid tails. These molecules are known as the cholesterol. These molecules are known as cholesterol molecules. As we discussed that the cholesterol comprises about 30% of the total lipids of the cell membrane. So between the tails of the phospholipid, which are made up of the fatty acid or which are hydrophobic or non-polar, there will be some molecules of the other lipid and the other lipid will be the cholesterol, which constitute about 13% of the total cell membrane. What is the function of these cholesterol molecules? These cholesterol molecules are important in maintaining the fluidity of the cell membrane. As we know that the cell membrane is not a rigid structure. If it was rigid structure, then no substance can gain entry into the cell or outside the cell. We know that on the regular basis many substances enter into the cell or outside the cell. This is the cell membrane. There will be the phospholipid bilayer, outer leaflet and inner leaflet. So we know that if the cell do wish to eat something like some bacteria, some virus, or phagocytose some cell, we know that the lysosomal cells do phagocytosis and it eats some foreign object which gain entry into the cell. So if there is some substance which the cell want to eat, it is present, the substance the cell wanted to eat, so the cell form this type of structure. Which part of the cell is form structure? This is the outer layer, this is the cell membrane which form this structure. Then this gain entry into, and then this vesicle which the cell membrane is formed will be removed and the phospholipid molecule are again lie. Initially the phospholipid molecule are arranged in this pattern. When it wishes to engulf some substance, then the phospholipid molecule will lie in this manner. Then it engulfs, there will be the tail, then it engulfs the substance and then again back the phospholipid molecule lie in this manner and the substance will gain entry into the cell. Same is will be the in case of cell wanted to remove substance. For example, this is a substance, metabolic waste. This is the metabolic waste, we can call this as urea. Cell wanted to escape it outside. So what will happen? This is the normal phospholipid bilayer. So what will happen that this substance come closer and the cell will also again form a vesicle like this, and this substance will be expelled out of the cell. And what will happen that the cell again will recover its cell membrane like this, and this substance is expelled out. So the substance, the process or mechanism in which the cell intakes or engulfs substance, this is known as the endocytosis. Cytosis means the process of eating and endo means inside the cell, and this is the exocytosis. This exocytosis is also known as the cell vomiting, because it resembles like that the cell is vomiting, because it engulfs, because it expels something, so it looks like that cell is vomiting. So we also call the exocytosis as the cell vomiting, whereas the endocytosis is the mechanism in which the cell eats something. The endocytosis may be of two types. This may be phagocytosis and this may be pinocytosis. We'll clear about the endocytosis, that in this process the cell will definitely eat something. In the phagocytosis the cell intakes some solid object, solid material, while in pinocytosis cell will intake some liquid material inside the cell. Okay, this both processes, the endocytosis, the engulfing process of the cell, and exocytosis, the expelling or vomiting process of the cell, both processes are known as the membrane biogenesis. These are known as the membrane biogenesis. So why the membrane biogenesis is occur? So the fluidity of the cell membrane maintains, as we discussed, that the cholesterol molecules are present. Cholesterol molecules are present in such a way that these are the tails of fatty acids, and these fatty acids are of phospholipid molecule. Obviously, this is the head of the phospholipid molecule, and these are the tail, and the head is polar and the tail are non-polar, as we discussed. The cholesterol molecules are intercalated between the tails of the phospholipids. And why is the tails are arranged in such a pattern? What is the purpose? So we know that cholesterol molecule also contain a hydroxide group at this region. Hydroxide group, there will be a hydroxide group. We discussed that in the ECF, extracellular fluid, or in the ICF, intracellular fluid, both are the aqueous environment, both contain the water, and water is a polar molecule, hydrophilic. So the hydrophilic heads are arranged outside the membrane, and in the same way in the inner leaflet, the hydrophilic molecules are also arranged in such a way that these faces the polar water molecule. So the polar attracts the polar, polar attracts the polar, and the hydrophobic attracts each other. Non-polar attracts non-polar. So these contain the hydroxide group. Hydroxide group is a polar. In the same way, there will be the hydroxide there, it is also polar. So in both sides, the hydroxide molecules are lying outside. Why these are lying outside? Because the hydroxide is polar molecule. So because of its polarity, it lies in the polar region, while the rest of the cholesterol molecule acts as non-polar, because we know that the cholesterol is a lipids, and lipids are made up of the fatty acids, and the fatty acids are non-polar. So this will act as non-polar, and that's why they are arranged in the non-polar parts. The tails are the non-polar, while the hydroxide ions are the polar. They are inside of the cell membrane or outside of the membrane, which faces the ECF, and the inside which faces the ICF. Both polar faces the polar, and non-polar faces the non-polar. So we discussed that why cholesterol molecule are arranged in such a way, intercalated between the tails of the fatty acids. Now, what factors will determine the fluidity of the cell membrane? So first of all, we discussed as about the cholesterol. So the cholesterol will definitely determine the fluidity of the cell membrane. How it will determine the fluidity of the cell membrane? We now discussed that the cholesterol molecules are present between the tails of the fatty acids. Means they are intercalated between the tails of the fatty acid. These are the phospholipid heads of the cell membrane. These are the tail. As I told you that it may be saturated or unsaturated. The unsaturated shows the dangling outside. These are arranged in such a way, and the cholesterol molecule are intercalated here, and the hydroxide ion is lying outside in the polar region. So more the cholesterol molecule in their path, more will be the difficulty for the phospholipid molecule to float in that region. While on the other side, if the cholesterol molecule concentration are low, this is the another phospholipid bilayer of the cell membrane, head and the tail, and these are, and in this cell membrane will show that the cholesterol molecule are present in the lower concentration. For example, cholesterol molecule are present here, and the hydroxide ion faces the outside polar end. Here and here only 2 cholesterol molecules. So you can see that here will be the 4 cholesterol molecule, but here will be the 2 cholesterol molecule. So the concentration of cholesterol molecule is larger in the upper region as compared to the lower region. So in the upper region, the fluidity will be lower. Why the fluidity will be lower? Because the phospholipid molecule will have lesser region to spread, so the fluidity will become decreased. While in the lower region, the cholesterol molecule not present consecutively, they will be present one after another or 2 after another. So the head and the tail will have a freedom to move, while in this region if it want to move here, then there will be the hindrance of the cholesterol molecule. Now there will no any here cholesterol molecule, so the tail can move freely here and here, but at this side there will be another cholesterol molecule, so there will be another cholesterol molecule, so the tails cannot move freely. So we now conclude that greater the concentration of the cholesterol molecule, less will be the fluidity, or vice versa, more lesser the concentration of the cholesterol molecule, more will be the fluidity of the cell membrane. So the cholesterol will determine the fluidity of the cell membrane. The other major factor that will determine the fluidity of the cell membrane will be the temperature. How will temperature affect? The second factor is temperature. We'll know that if the temperature increases, then the molecular motion or the kinetic energy of the molecules increases, and the because of the increase of the kinetic energy, the molecule will move faster. We know that this relation we studied in physics, that temperature is directly proportional to the kinetic energy. So we have to draw the cell membrane. This is the phospholipid head. These are the phospholipid head, and these are the tail, which may be saturated and unsaturated. Unsaturated show a process outside this way. Then if someone increase the temperature, if the temperature is increased, then these are the molecule of the phospholipid. So the kinetic energy of these phospholipids molecule will increase, so they will run faster. Here, this direction, they will run here fasterly. Suppose their initial speed will be 5 meter per second, but due to the increase in temperature, their speed will become double, 10 meter per second. Why? Because due to the increase in temperature, the energy of the phospholipid molecule will become increased. Because the energy increases, so they will increase, they will move faster, and because of the faster movement of the phospholipid molecules, the fluidity will definitely increase. So we can say that temperature is directly proportional to the fluidity of the cell membrane. Greater will be the temperature, greater will be the fluidity. While we can also say that the concentration of cholesterol molecule will be inversely proportional to the fluidity. Greater the concentration of the cholesterol molecule, the lesser will be the fluidity, or lower the concentration of cholesterol molecule, the greater will be the fluidity, because they have enough space to move, to spread, to move freely. So the third factor is the saturation of the fatty acid. This will be the saturation of fatty acid. We are talking about which type of fatty acids and which saturation. So let's take a quick review of the phospholipid molecule. We discussed that phospholipid molecule has two regions. This is the head, this is the tail. The head is the hydrophilic, means polar, it will dissolve in the water, while the tail is the hydrophobic, means it is non-polar, it will not dissolve in the water. And in the structure of the tail, we discussed that why the tail is hydrophobic, because of the fatty acids, while the head is hydrophilic because of the phosphate, and the phosphate group is polar. So the tail, we discussed that in one phospholipid molecule there will be the two tail, and the two tails will be saturated, and in case of the saturated, they will lie as straight in this way, in this posture, while the tail will be unsaturated. Both the tails will be unsaturated, or one tail will be unsaturated. For suppose this tail is the saturated and this tail is unsaturated, the unsaturation of the tail is shown in such a manner that the one tail is dangling outside, while the other tail, which is saturated, is lying in such a way, in a lining manner, while the this tail shows the saturated fatty acid, in which there is no any double bond, while this tail, which is dangling outside, is the unsaturated fatty acid, in which there may be a one double bond or two double bond, three double bond, as we quoted earlier that oleic acid is a monounsaturated fatty acid. So saturated, in the case of the saturated fatty acid, what will affect the fluidity of the cell membrane? So greater will be the saturation of the fatty acid, lesser will be the fluidity, while greater the unsaturation, greater will be the fluidity. So we can also say that the saturation of the fatty acid is inversely proportional to the fluidity of cell membrane. We can see this Suppose this is the cell membrane, phospholipid heads, these are the phospholipid tails. In this phospholipid tail, I'll draw only one tail which is the unsaturated, while the all other tails will be saturated. So you can see that in this phospholipid molecule, the tails are more saturated than the unsaturated. So here is tails are more saturated, while another model I'll draw about the tail in which are more unsaturated. These are the phospholipid heads, and there will be the tail definitely. I draw this unsaturated, this also unsaturated, this is saturated, this is unsaturated, this is unsaturated, this is saturated, this is unsaturated, saturated, unsaturated, and unsaturated. So this is more saturated model, and this model of the phospholipid will represent the more unsaturated tails of fatty acids, tail of fatty acids. So what will happen? How the saturation decreases the fluidity and how the unsaturation of the fatty acid increases the fluidity. So we can also call this tail as the leg of the phospholipid. These are the legs, and we can also call that these cholesterol molecules are intercalated between the tails or the legs of the phospholipid molecule. So these legs are dangling outside in this way. If these legs are dangling outside, then more legs, if they will dangle outside, then there will be the more motion, there will be more motion of the phospholipid molecule, which will result definitely in the increase in the fluidity. But if these are in this way, this straight, then there will no any more motion than the as compared to that a model in which the tails are more unsaturated, while the lower model in which the tails are unsaturated, they are like this and the other is like this. So they will more move, there will be more unsaturation, there will be more movement, and because of more movement the fluidity will increase. So we can also say write in this way, we can also write in this way that unsaturation of fatty acid, unsaturation of fatty acid. Which one fatty acid? The fatty acid which comprises the tail of the phospholipid molecule is directly proportional to fluidity. Why? Because more the unsaturation, more will be the dangling of the legs, and more will dangle the legs, there will be more motion of the phospholipid molecule, and because of the more motion the fluidity will definitely increase. So that's about the fluidity of the cell membrane.